So I guess we'll go ahead and pick up where we left off yesterday. So yesterday I went over what raw RNA-seq data is. So if you guys recall, after you sequence your libraries, you then have FASTQ files which contain reads, and each read is a set of 4 lines. And so these 4 lines we went over yesterday. So it starts with this header line, which has information about the sequencing run, such as the sequencing platform, run number, flow cell ID, location, read number, and the index. And some of you guys noted that this one here is indicating what number read that is. So this is the first read, or our forward read coming off the sequencer. Some of you guys also noted correctly that there is one index here. So we only have one set of this, and this index is 6 base pairs long. And one of the things I want to point out, because I know there was questions about paired end reads, so how the paired end reads are connected together in these algorithms is they'll all have the same exact read header information, because this information also contains the clusters that they were in, right? So the forward and reverse read are going to be coming from, of course, the same cluster, and so it'll have the same information here. The only difference is that this one will be different. So it's usually a 2 there, not always, but usually a 2 there. To indicate that it's the reverse read from the forward one. And then just to finish with the overview of what a read looks like, so line 2 has our nucleotides in it, so that's the actual sequence. Line 3 is a spacer line. Sometimes it starts with a plus sign. It usually starts with a plus sign, but it could also just be entirely blank. And then our 4th line is our quality scores of each base, and these are encoded in a FRED 33 using ASCII 2 characters. And I just showed you quickly before we left yesterday that this is what those quality scores equate to. So it's the Q scores are on the left of each one of these columns. In the middle is the error that represented by that Q score. And then on the right is the ASCII character that corresponds to that Q score. And so just to kind of interpret these here, so if you have a Q score of 10, which is represented here, you have an error rate of 0.1 or 10%, which means that your base call accuracy is 90%. And then just going down the line, so if you have a Q score of 40 over here on the right, that's going to be equivalent to a 0.0001 error rate or a 0.01% error, which is going to be a 99.99% accuracy in your base call. And so pretty much to just quickly take a look and interpret the quality scores from the reads. Basically, anything that has a high quality score is represented by a letter, whereas anything with lower quality scores you'll see is represented more by symbols. And so just to take a quick look back here, you'll notice that the majority of these calls had a quality score of J, and J represents here, so 0.00008, probability of it being wrong, or 0.008% error rate for J, so really high quality. Of course, these FASTQ files will contain millions of reads depending on how much your read depth is, and so it would be really hard to try to assess the quality of all the reads in your samples by looking at it manually. So thankfully, there are tools that have been created to do this for us. So FastQC is probably one of the more common tools used to assess the quality of reads inside FastQ files. And so if you recall, bring it back to the workflow here, you recall the RNAseq workflow, the first thing that we do once we have our raw reads is we assess the quality. And so we're looking at the quality of each read, and we're going to use the FastQC tool to be able to do this. And so when we run it through FastQC, we'll be able to assess the quality of sequence data for all the sequences of a sample, and it'll give us a nice HTML report, a file that kind of summarizes the quality of all of our reads. And so one of the first measures that it has here is just some basic statistics about your read quality. So it'll tell you what file name it was looking into, the type of file, the base call file, the encoding that was used, so it can identify that it was Illumina, and Sanger. So the quality of the Fred 33 scores, those are actually the same scores that are used, same algorithm used in Sanger to identify scores. So that's why you see Sanger and Illumina. And then total sequences. So just going to prod you guys for this. So what, I guess I have a couple questions here. We have sequences flagged for poor quality, sequence length here, and then %GC. So I'm just going to have you guys take a look at the information here. And I first want to ask you, are these data forward or reverse reads? Just curious if you guys can gather that from here. Guesses, you can unmute your mic, put it in the chat. Reverse. How do you know that, Ty? Mark, you guess forward. From the R2, yeah. So in the file name, you'll see up here that there's an R2. So I don't want you guys to get too used to this because most bioinformaticians, when they're preparing their file names, you will, and coming off the sequencer or coming off the BCL to FASTQ algorithm from Illumina, they'll have a 1 or 2 some indication of the read number in the file name. However, people can change file names. And so most bioinformaticians will make sure in the file name, if it's paired end read, that it'll indicate either R1 and R2 or just a 1 and a 2 or something like that. However, that's not always the case. So although we do see that here, you can't go by that exclusively to know if this is forward or reverse, but in this case, it's true. I made this this name, so it has R2 in the file name, so we know that this is a reverse read. What's the read depth for this sample? How many reads are in this file? You guys could type in the chat, unmute your mic. $150 reads? Jill, why do you think it's 150? Say a lot. Yeah, exactly, Jill. So that 150 is referring to the sequence length. Yeah, YGen, you're absolutely right. So YGen said that it's around 100 million. YGen, how do you know that? Total sequences, yeah, exactly. So if you see this measure of total sequences, that's telling me how many reads are in this file. And so as YGen noted, that there's over 100 million reads. So 100,600,211 reads to be exact. Just a quick note here. So if it is about 100 million reads, you don't have to tell me the exact number, but what calculation would you do to know how many lines you would see in that file? So remember from the previous slide, each read has how many lines?
Multiplied by 4.
Yeah, exactly. So if you were to multiply this by 4, you'd get the total number of lines in that file, right? Because each read consists of the 4 lines that we just went over in the previous slide. This is helpful. So remember how I mentioned that in flow cells you have multiple lanes. So sometimes when you get your FASTQ files back from a core, they might not have put all of the sequences from each one of those lanes together. And so they might give you 4 sets of FASTQ files per sample. And then you'll have to combine them together with the cat command that we went over in the intro Unix, and you can kind of check the line count with the command that I didn't go over, but we can call WC with the option -L that'll count the number of lines in a file. And so I usually do that when I have to combine files together to have one set of FASTQ files per sample. And so I usually do a check on how many lines were in each file before I concatenate, and then after I concatenate it to make sure everything was combined together correctly. And so you can know the number of sequences that you have based on the number of lines, just dividing the number of lines by 4. So that's why I wanted to bring that up. And then Joel, you kind of jumped the gun on this one. Amanda, why R2 and not just R? For the labeling here?
Yeah, I mean, you have one forward and one reverse, but you called it R2.
Yeah, good question. So some people do use F and R to indicate forward and reverse. In the notation that I used here, that R stands for read. So
Right.
read
But
one, read two.
oh, it's the second read. It doesn't necessarily reverse when you use R.
Right, right. Yeah.
Ah.
So, but some
Mm.
people do use that notation, as Mike noted in the chat. So again, you can change file names. Remember how we use the move function to rename files? You could do that to any file. So bioinformaticians will rename files, and scientists can rename their own files too. So you can change the name to whatever you want it to be. And so people will use different conventions to indicate forward reverse. So you can use F for forward, R for reverse. You can use R1, R2, which is read one, read two. For forward and reverse, you can just use one and two. And some people won't put anything, which can be really frustrating. So I'm already put this out here just because Joel jumped the gun and answered this with the read depth question, but the sample read length for us is 150, and we know that because it tells you sequence length 150. So again, really nice. And you'll also notice, right, these are raw data, so the sequence length is only one value. So that also gives me confidence that this is indeed raw data. Nothing has been done with these reads yet. The next thing that the FastQC tool will give you is the quality per base. And so down here on the x-axis, we see the position in read and base pairs. And so because these are 150 base pairs long, we see that it goes from 1 to 150. And then this gives us that Q score. So the quality score here. So if you remember, a quality score of 40 is 99.99% accurate. And so it also, the FastQC output just gives some color coding for what they consider really high quality. It's in green here. Q scores of 28 or higher, they're considering really high quality. They're considering moderate quality, so good quality between Q20 and Q28, and then they're having here in red poor quality, anything less than 20. So a couple of questions about the quality is, do you notice, and I should say that this is looking at all reads in the FASTQ file, right? So that's why these are box and whisker plots. So you're seeing the quality of all reads for each position of the read in the file. And remember, there's over 100 million reads in this file. So this is a summary of across all 100 million reads. And so do you notice anything about the quality as you move towards the end of the read?
Yeah, they get worse as you go further along.
Yeah, why do you think that is?
Well, a lot of enzymes do that.
Yeah, yeah, yeah.
That's a feature of enzymes. This isn't exactly the same, but I don't know if it's an enzymatic, but there's more diversity at the 3' end of sequence than there is at 5', and the thinking is that's enzymes kind of not being able to function as precisely as they go down the sequence. But I don't know if that's the reason here.
Part of it. So it could be the reason, right? So, and I think of it as it more so like our cells
Could
are
it be
very. Oh, go ahead.
could it be actually on the side of the machine? Maybe like as it's going along, you lose integrity of its I don't know how to put this in words, but like its read. And as it goes along, it's kind of like probability. It loses its integrity over time. So it's actually on the machine side of things. I don't know if that's anything close to being correct.
Kind of more so on the substrate side of things. So both, if you think about it, right, our cells are wonderfully suited to for the perfect environment for polymerase to work optimally, right? So polymerase is the enzyme that we're using here to synthesize our sequence in the sequencing by synthesis reaction. And so we haven't quite made conditions as ideal as they are in our bodies. And so the polymerase will work for a while. It'll go through and it'll sequence your read as it goes, but it's going to eventually fall off. So if it does fall off, you'll tend to see poor quality at the end of the reads for that reason. So that was what Elba was alluding to. And then to your point, Chief, remember each of these nucleotides are tagged with fluorophores. And so once you put those fluorophores into the reaction, right, that they can start to degrade over time as well. So you might not get as strong of a signal. The other reason is depending on the diversity of your library. As you're going, right, it's incorporating the base pairs as it goes. So we also have that blockage at the end of a base pair to make sure that only one base pair is incorporated at a time until a picture is taken, and then that termination is removed, so that way the next base pair can join on. And so over time, as these reagents are in the machine, that blockage could come off, and then you can have two nucleotides incorporated at a time, which will confuse the signal. So which is kind of like an instrument, but really more a substrate, a caveat of the substrates being in there over time. So we do tend to say it's pretty common for all those reasons to see the quality kind of drop off at the end.
So Amanda, is it worth it to get 150 reads if the tail is ambiguous in quality?
Good question. If you take a look, even though the quality is worse at the end, the majority of those reads still has quality that's in like insanely good quality, right? So it's still, I think 30 is what, like 99.9 something percent accurate. So even though it's less than at the beginning, it's still really good quality. And so you do have some of these outliers, which to your point is, remember, the next step in the pipeline is for us to trim and filter our data. So that's what we're going to account for in any reads that have like really poor quality, kind of in the red here, data. We can actually just lop off the end, so that way we don't consider it when we do our alignment. Good question.
And what about the quality at the beginning? Is that just the enzyme binding? It's like
Yeah, kind of the enzyme hopping on. You usually do see a little bit of wobble beginning.
Yeah.
Sometimes we'll notice in
It is
some other
wobble.
notes too, that it's just not as clean base call right at the beginning. The other question that I have here are, these data are for the reverse reads, and do you think the quality would be different for the forward reads? Any guesses?
If it's chemistry, I would hope not, but maybe.
Yeah. So you would think not, but the quality of the forward reads is often better than the reverse. We're not entirely sure why. Some hypotheses is that, right, you're starting a reaction and we sequence the forward read first. And so remember, all of these reagents inside our flow cell are going to be kept at -80. And then we thaw them and use them. So we go through a thaw cycle to use them in our instrument. And as the instrument is going on, right, the temperatures within the flow cell are going to be changing for the denaturation process and things like that. And so one hypothesis of why usually the quality of the reverse reads are slightly lower than the quality of the forward reads is because the reverse reads are sequenced last. And so all of those reagents have been in there exposed to room temperature at least for that length of time. And so maybe the efficiency of some of those substrates and enzymes reduce as it's continued to be exposed to room temperature. So that's kind of one hypothesis about why that is. And we'll see that in the data when we analyze it in our Jupyter notebooks. So the next thing here is per sequence quality scores. And so this is another way to look at the quality scores, whereas before the previous slide here, it was per base pair. So you saw the position in base pair was here on the x-axis and quality on the y-axis. Now we have quality scores on the x-axis, and here is just total number of reads on the y-axis. And so what it's basically showing you is how many reads have a specific quality score. And so it's just another way to look at and interpret the quality. And so what we see here is the majority of our reads are going to be in this high quality range. So kind of gave you this answer, but would you consider these data to have good quality overall? Yeah, Mark. Yeah, Ty, yeah, really good quality here. And you tend to see that with Illumina. Again, there's a reason Illumina won. Their sequences tend to be really high quality. And if they're not really high quality, sometimes it can be an instrument glitch. And actually, at least in our experience, we have had that happen where we see something weird with the flow cell and the sequences coming off, and we'll actually contact Illumina. And oftentimes they'll replace a flow cell if something goes wrong with the machine. because usually we see quality that's this good from Luma Instruments. Next is a per base sequence content. So it's really good to have a very diverse library, and this is in order to interpret the base calls. So if it's diverse, then you're not having the same wavelength over and over that could confuse the machine. You should be detecting the different wavelengths for the different base pairs. However, if you notice here, these values, so we want these values to be pretty consistent across the read. So this is position in the read again on the x-axis, and this is percentage of the total reads being ATCs or Gs, right? So if it's a very diverse library, we should see all of these hovering right around 25% each across the positions. If the library is biased, you would see a spike in these data, right? So if the library was biased, it wasn't diverse, and maybe like in position here, we have a high number of A's or something like that. What you do notice is at the beginning here, we do see what looks like some bias, right? Any ideas why that is for RNA-seq data? Oh, I just gave you the answer, my bad. I'm gonna reverse that. Why do you guys think poly A tails? That's a good guess. It could be, but really for a poly A tail, right, I wouldn't necessarily anticipate that it would only come at the beginning. If we had, like, for whatever reason, a bunch of poly A tails in here, I would assume that there would be, if it's poly A tail, right, we would have multiple base pairs as poly A. So I would assume that it'd be more than just in positions like 1, 2, 3-ish. It would probably be further, and then we would see it be A. So here you notice this spike here is actually G. It would be A or T. So good guess. Low sample size. So what do you mean by low sample size? So we're looking at reads from one sample.
Oh, I mean, low number of nucleotide because they are only on the first one.
Low number of nucleotides. So, well, low diversity. Is that what you're referring to? So low diversity is what we have here.
I don't know if I read this chart correctly. I thought the first one is position, right? So
Yes.
you're in the first position, so I thought the number going to be even out gradually because you're talking about the total percentage of the sequence.
So yeah, here's total percent. So this would be what's the percentage of A, T, C's, and G? It's at position 1 of all the reads. And so if we have a very diverse library, position 1 at all the reads, right, should be randomly distributed, which means that it should be roughly even A, T, C's, and G's, unless there was some bias in our assay to cause that first base pair in over here. We got 45% of the reads where the first base pair is actually a G.
Mm.
And so I'm curious to know why would that be the case?
We're not reading any part of the primer adapter or any of that, right?
We could be. Oh, not on this side. Sorry, that was
Not
the
on
other
this
side of the
side,
read. Yeah,
no.
not at the beginning of the read.
Because it should be, equal amounts G and C and A and T, and
Right.
it is.
Yeah, you would
Except
think.
at that beginning part, which means that there's a bias and it looks like they're somewhat parallel, but not exactly. You would think.
Yeah.
Yeah.
So Ty, you're right on, but I'm going to follow up with that. Mark, primer diamond contamination, that could also be the case, and we'll see that there's actually another graph that looks specifically at that, so that could also be the case. But Ty, what primers are you referring to? Yeah, Ty's got it. So remember when for RNA-seq data, we start with RNA, right? And our first step in library preparation is to convert that RNA. Well, actually, it's our second step. Our first step is to remove ribosomal or enrich for poly A, and then to chop it up. But after we chop it up, our next step is to convert that RNA into cDNA. And so when we do reverse transcriptase, converting it to cDNA, we use a kit that uses random hexamers in the priming of the RNA to convert it to cDNA. And presumably, we hope that those random hexamers are unbiased. But in reality, nothing's really unbiased. So there is some bias in the priming of the RNA when we're converting it into cDNA. And so because of that bias, we see it reflected in our sequence here for that little wobble at the beginning that we have. But that's going to be at the beginning of the read, and then we'll see it kind of even out as you get into the read. So yeah, nature of RNA-seq primers cause some wobble at the beginning of the read. The next thing that we see in our FastQC data is the per sequence GC content. And so across the x-axis here, we have the average GC content in percentage, and the y-axis is going to be total number of reads. And so what you would assume is that we would see some sort of normal distribution. And then that percent GC normal distribution is going to be the percent GC of the tissue that you're looking at, or the organism that you're looking at. And so we would assume that it would be a normal distribution, which is depicted here in blue. The actual GC content for our sample is in red. So we see, I think I have some questions here. So yeah, we expect the GC content to be normally distributed, so similar to the blue line. And so the question is the GC content normally distributed across all reads in these data? What do you think? Yes, no, no, yes, no? Anyone else want to weigh in? Do you think the red's normally distributed? So I have here kind of. Yeah, Ty, kind of. So the reason, so Mark, I could see why you would say no, right? Because we do have this kind of little blip over here where there's some, what is this, like 500,000 reads or so have kind of high percent GC content, which we're gonna get into in a little bit. And then we kind of see up here that we got some spikes too. And so, but in general, this red kind of follows the normal distribution with these spikes. With some little spikes here. So what do you think the peaks outside of the normal distribution could indicate? So why do you think we might have some peaks outside of our normal distribution? Any guesses? Genomic island, yes. Good guess. Technically transcriptomic because we're looking at RNA. But yeah. Any other guesses? So it actually indicates some form of contamination usually. So anytime we see a spike here, that means that we have some sequences that have high GC rich levels. And so that could be because we have a repetitive sequence. So maybe a transcript that was transcribed from a genomic island, which has a lot of repeats in it of G's and C's, which could account for that. It might be adapter dimers, depending on the GC content of the adapter dimers we might see. It might be an indication of some other, like a bacterial contaminant, like especially if you're thinking about if we're processing tissues. As you guys know, as biologists, humans, animals, we have more microbes than we do our own cells. So there's going to be microbial RNA in here when we do our RNA extractions. And so especially when we do total RNA, of course, we wouldn't have that treated poly A, but so it indicates some form of contamination. And I'll tell you, I think in the next slide, no, I don't have it here. So I don't have it listed here, but we will see it in the sheet that there is also overrepresented sequences that are given to us in the FastQC analysis. And so sometimes if we see some of these peaks in our GC content, we can look at the overrepresented sequences that FastQC notices, and we could do like blast searches on them to kind of try to identify what those could be. But honestly, this actually looks pretty good. I'm going to get into a little more of this bigger GC blip here and what it could be. That has to do with the chemistry of the sequencing, likely. The next graph we have here is it says the per base N content. So anytime the Illumina instrument, when it's doing the sequencing by synthesis, is not confident in a base call, it will report an N. And so a couple of questions here. Oh yeah, I said that. So things are cannot distinguish the base call and N is recorded. How often was a base unable to be called in these reads? A lot, a little. Yeah, very little. So you see, this is mostly, again, we have the position and base pair across the x-axis, and here is the percent of total reads. And so we see this little blip here at the beginning, maybe like 1-ish% at the beginning we get an N, and that has to do with the stuff that Elba was talking about, you know, the polymerase kind of latching on, maybe that first base being kind of difficult to interpret or distinguish. But it's incredibly rare to see an N. And this, again, is pretty consistent with Illumina technology. You usually get pretty clean data as long as you have good libraries going in.
So Amanda, is the detection, because the base call is made on some kind of signal to noise ratio fluorescence detection parameter, right? This is this base, there may be other fluorescence. So is that a parameter that you can set? Is that something that the person who's running can manipulate that parameter? Or is that detection of the base call, is that done by an algorithm?
Yeah, the detection based call is done by an algorithm based on, right? So you take an image in each one of the wavelengths that you're recording. And so, especially for that first call too, right? It's also going to be, so it could be instrument detection. There is going to be, you know, maybe some background in there at the beginning. We have, so at least for Illumina, they have their technicians come in, set up the sequencer, and do some initial runs to kind of normalize and identify what the background subtraction and stuff should be inherent to the instrument. And so we use those settings, but you can, I'm pretty sure, manipulate some of those settings yourself if you want to. It's not recommended by Illumina. So usually if we start to see for some reason, like a lot of N's being called or something, like 1% at the beginning is not a lot, and it's fairly common, so it's really nothing to worry about. But if you do tend to see these like getting higher, then that's when you would want to call your Illumina rep in our case, or whatever company made your sequencer, and tell them to come over and recalibrate your machine. So the next thing that we see from the FastQC analysis is a sequence length distribution. As you guys noted from the statistics summary at the beginning, all of these are 150 base pairs. And so, again, just making a note that the length of all raw reads should be the same because, right, we control that. We set the number of cycles that the instrument does, and so we should get back that exact number of base pairs for every single read. And then we kind of already answered this, but yeah, all the reads are the same length that we noted in the basic statistics. There's only one value there, and here we see everything is at the 150 base pair length. We already know this, 150 base pairs. So the next thing that's super useful and can get to your point, who is it, Mark, that asked about the primer dimer in terms of that GC contamination. So this is where we can start to look to see if there is adapter content in there, right? So if we do have adapter dimers in here, we should be able to detect them in the FastQC. So FastQC has inherently built into the tool, it has a list of the adapters that are used by common sequencing instruments. So you'll notice a bunch of Illumina instruments and kind of older school Nextera and Solid adapters too, which are different sequencers. And you can see here, nice and convenient, that I don't want to answer. So if your sequence of interest is shorter, so just why we would have adapter dimers. I know we talked about this when we were talking about library preparation, but these can occur if your insert is shorter than the number of cycles that you programmed the sequencing instrument to sequence. And so let's say if you're sequencing at paired N150 and your insert is 100 base pairs. So if that's the case, you're still going to sequence 150 base pairs, but now your insert, your sequence of interest is only 100 base pairs. So after that 100 base pairs, it starts to sequence into the adapter. And so that's why that's one reason that we can have adapters in here. The other reason that Mark mentioned is it could be adapter dimers that are in there as well, and that would also be reflected in here. So a couple of ways we can give adapters. Does this example contain adapter sequences? Yes, no, no, yes. Yeah, this does. And so we know here, we could see that as you get to the end of your base pair, right? Because it should sequence your insert, your region of interest first. And then if it's shorter than the number of cycles, then the cycles will continue into your adapter. So you'll only see the adapter contamination at the end. Unless, of course, you have adapter dimers. So that's another thing I should point out, Mark. So if we do see adapter dimers, we would also see adapters at the beginning. So luckily, we don't see much at the beginning. So we probably don't have adapter dimers, or at least not that many in here, which is a really good thing. So that answer is yes, it does, because we see that as you get to the end of the read, we see the adapters come up here. Oh, I already answered this, but where are the adapters relative to the read position? I already gave you that answer at the end of the read. And do you think this, well, I guess I should ask first, what adapter do we see here? Notice it based on the key. Universal, yeah, so specifically Illumina Universal. So we see Illumina Universal adapters, and those are the adapters that we use at GeneLab, and they're probably the most common adapters used in Illumina sequencing, unless you're doing a specific type of sequencing for different molecules. But yeah, so we see those Universal Illumina adapters. And do you think that this is going to be a problem for our processing if we have adapters in there? Not if the majority is a sequence. Yes and no, depends on your mapping rate. You can trim those seeks, right, yeah, you can, Ty. All right, so this is kind of where looking at the quality of the raw data is how you're going to determine if you want to trim, right? So looking at the quality, as we noticed at the end of the read, we saw that quality for some outliers dip below into that poor quality range. And then here we're seeing that we have some adapters. And so this could be a problem. And the reason is, right, so we don't know where these short sequences came from yet, right? So we ultimately want to take these short sequences and map them back to our reference genome. But remember, these adapters we added on artificially. They didn't come from biology, right? We added them on. And so now when we want to map this full read back down to the reference genome, if we have, the last, let's say in some cases, 50 or so base pairs that we added on artificially, it's probably not going to map to anywhere, at least not anywhere correct on the genome, because we added them artificially. It didn't actually come from our biology. And so that's why it could be a problem, and that we might not want to have this. And depending on how many mismatches we allow when we do our mapping back to the reference, we may or may not be okay with the majority of the sequence. So Mark, to answer your question. So it is always good, as Ty mentioned, to get rid of these before we do our mapping. The next thing that FastQC evaluates is duplication levels. So I do want to note some things about duplication levels. I know we talked about them a little bit. We're going to talk about duplicates a bit more in the next slide. So this is how FastQC evaluates duplicates is it only looks at the sequence of interest, right? Or sorry, of your read. So it looks at the sequence on your read, and it only looks at the first 50 base pairs of that sequence. And it also only checks the first, I think, 200,000 reads. So it's really only taking a sampling to see if there's duplicates. And the reason is to save processing time, right? Because if it were to go through and look at the entire length of every read and do an alignment analysis with every other read in there, when you have 100 million reads for this example, it could take a really long time and a lot of computational resources. So how the FastQC algorithm got around that is it kind of takes a subset of your data and does the evaluation based on that subset. So I always take the FastQC duplication levels with a grain of salt. They're usually overestimated in here. And so, yeah, the purpose to use to determine how unique the reads are. Again, it has 2 lines here in this. So the blue line is going to indicate the percent of duplicated reads at a specific duplication level. So the duplication level is here on the x-axis. So over here, 1 means that there's 1 copy of the particular sequence. Here means that there's 2 copies, 3 copies, 4 copies, you get it. And then this is the percent of overall sequence. The percent of all sequences. Again, it's not all the sequences that we have, it's just the percent of the ones that this algorithm evaluated, which is only about 200,000 reads. The red shows what these data would look like post deduplication. So if you were to run deduplication just based on the read sequence itself, what you would see is what's in the red line. So reduplication, right, can be either biological or technical in origin. Do you guys remember how? We could start with biological. How do you get a biological duplicate?
Gene duplication.
Yeah, or like overexpression of a transcript, right? So if you have like multiple copies of a transcript in your original sample, if something's really highly expressed in one of your samples, although we try with the divalent cations when we do our chopping of our mRNA, we try to make that should be, as random as possible, but it always going to have some biases. So if you have the exact copies of a transcript, right? So really high expression of a transcript, when you do the cutting up of that transcript, you can chop at the same location on each one of those transcripts. And if that happens, then you'll have reads from different copies of the transcripts that are identical, but it came from biology because that particular gene is really highly expressed, so there's multiple transcripts, and so those reads are true biological duplicates. We can also get technical duplicates. There's a few ways you get technical duplicates. I went over some of them. Does anybody remember how we can get technical duplicates? So not originating from biology. Anyone remember? Think about library prep. What's the last step that we do in library prep? Yeah, Ty, amplify. So the last step of library prep, remember, we go through and we amplify our libraries. So we go through and we run them through a few cycles of PCR to make sure we have enough material for sequencing, but that also introduces artificial duplicates into your libraries, right? And so we can have technical duplicates that were derived because we made copies of all of our libraries when we did that final PCR step. And so that would be an artificial duplicate because we did it in a machine. It didn't come from our original samples. And so those are technical duplicates. So if you have a very diverse library where all the reads are unique, you would see 100% of the reads would just have a duplication level of 1. It almost never happens. So it's common specifically when working with enriched libraries, right? So enriched being the fact that we have PCR amplification, have some level of duplication. Why? I guess I already answered that question. Because they're artificial copies that we're making during that application step. And then just some discussion about the types of duplication levels, right? So we see here in this, in our sample here, that we have 20% of the reads sampled, again, of the like 200,000 read sample, and only of the first 50 base pairs of that read. So remember, these are 150 base pairs long. So we're really only looking at a third of the read, and we're only looking at 200,000 reads in total here. So we see, based on that subsampling, about 20% of them have one copy. We see what about 8% of them or so have two copies. We see about 3% of them, or about probably about like 4% of them have three copies, and etc. So that's how you would interpret this. And so we see some peaks here towards the end. So we see that roughly 10% of the reads that were sampled have 10 copies or more. And then it kind of goes on like that. So where these peaks are for the number of copies of your sequence kind of tells you a little bit about the type of where those came from. So lower duplication levels in like the 2 to 9 range could be reads derived from interesting genomic regions like mRNA. So that might account for some of your true biological duplicates that we were talking about. Moderate duplication levels in the 10 to 100 copies could be reads derived from ribosomal RNA. So if we have some ribosomal RNA contamination in here, which can be highly repetitive sequences, right? So we might see that those exact copies came from ribosomal RNA, or other highly repetitive genomic regions could cause us to have sequences that are identical. High duplication levels that are like over 100 copies can be indicative of a library issue. So that could be indicative of adapter dimers, right? Because adapter dimers are all going to be the same sequence. If you spike in, when you do your spike in with Phi-X, if you add a little bit too much, Phi-X are going to have similar sequences, or it could be some other contaminant that you have in there. And so, question here, are duplicates present in this sample, and should we be concerned? So are there duplications present in our sample? Yes, duplicates are present, right? Should we be concerned? Is unsure. So I have yes and yes. So there are duplicates present, as Ty indicated in the chat, and indicative from this blue line here, right? We see that there are percentages of these subsampled reads that have duplicates. Again, caveats here is it's not distinguishing between biological and technical, right? So if these are biological duplicates, then there's nothing we should be concerned about. However, what I get concerned is when I see kind of these higher, the duplicate levels of like over 10,000 reads that are all exactly the same, and it looks like in the subsampling it's like almost 20%. That might be like an adapter dimer or something like that, which would concern us. And that's where we kind of can look at some of these other graphs to know what it is. And then we also have, which I don't have on here, which we'll see in our, when we run this in our Jupyter notebook, is it does give us overrepresented sequences. So then we could start doing a blast search on those overrepresented sequences and see what they are. There are ways to distinguish between biological and technical duplicates, as I mentioned when I was going over the types of adapters that we have. So for GeneLab, for instance, we have a UMI, unique molecular identifier, on the adapters that we use, and that UMI is added before we go into the PCR step. And so that means that the unique molecular identifier, if you have the same exact sequence and the same exact UMI on multiple reads, then you can be more confident that that was indeed a technical duplicate that came during the PCR amplification process. So yeah, I would be concerned here, but again, I always take this with a grain of salt because of those caveats where it can't distinguish between the two. It's a very small subsampling of all of your data. And so you really want to do something like a UMI assessment to really know what your true technical duplicate rate is. Then I just have here roughly what percent of reads, what percent of these subsampling of reads are duplicates. Tells you at the top of the graph. what they would be if they were deduplicated. Any guesses? What percent of the reads, at least from this graph, are duplicates? Okay, I'll just tell you. Yeah, roughly 75%. So about 73.5%. So basically here it says that if you were to deduplicate this, based on what they're telling us, that it'd be 26.58% of sequence remaining, which means that 100% minus that percent, about 73.5% are duplicates. Again, subsample, so this is probably an over representation of the actual number of duplicates in your full sample. So talk a little bit more about some other ways to get technical duplicates. So a couple of things, and Elba, I think you brought this up yesterday in terms of the clustering and how sequencing is done. So a single cluster that has falsely been called is 2. So this can happen. There are 2 types of flow cells that Illumina provides. One is an unpatterned flow cell. It looks like this, and that's also the flow cell that was depicted in that sequencing by synthesis video that we watched yesterday. And so in an unpatterned flow cell, we just have oligos in certain positions on the flow cell. There's nothing really distinguishing one set of cluster from another besides just a little bit of space. So it can happen where, especially if you overload your flow cell, where you have one of your inserts kind of latching on to a probe on the flow cell and folding over, forming a cluster, but then also folding over into where another cluster is and kind of forming 2 clusters that are close to each other. And so when the algorithm is interpreting these clusters, even though this all came from one original insert or one original library, the algorithm could think that it's 2. And so then it would consider this 2 separate clusters, and they're identical, right? Because they came from the same library. And so then that would be called 2, and that would be a technical duplicate. This only happens on non-patterned flow cells. So one of the ways that Illumina went around this is to make these patterned flow cells. So you see these little dimples here on this flow cell, and these are the flow cells that we use at GeneLab. And so in each one of these little dimples are oligos. And so the goal here was to kind of minimize the likelihood of this happening. And so you would have, when one strand of your library comes in here, it'll populate and fill up this entire well, and it would only be restricted to that well. And then you would have that for each one of these little dimples, right? It would be its own unique cluster. However, it can happen where you have one of your libraries being attached onto here that also folds over into another dimple, and then will be the same library will populate both of those wells. And so that could happen, and again, that would be a technical duplicate. And so that one's unique to patterned flow cells. And then we have ones that are present on both unpatterned and patterned. One is the PCR duplicates that we talked about. So an artificial technical duplicate from that PCR amplification step, meaning that you artificially made copies of individual inserts or sequences. And so you can have a copy of each one of those artificial copies made being populated in a cluster. So that would be a technical duplicate. The other thing is complementary strands from the same library. Remember, our libraries are double-stranded, right? DNA likes to be double-stranded. So the first thing we do is denature those libraries before when they're loaded onto the flow cell. And so, of course, now you have the same, like, one copy of it. When it's denatured, there's going to be 2 there, so you can have your sister complement strands each populate a well, and so that would be coming from the same sequence. So there are a few different ways that we can end up with technical duplicates. Questions? Okay. So that brings us to the end of assessing raw read quality. So as you guys noticed, There were some adapters that we identified in our reads. There were also some kind of outliers with low quality base pairs towards the end there. And so after looking at the raw quality, after looking at the quality of the raw reads, we can be confident that we probably want to trim and filter this a little bit to clean it up before we align it back to our reference genome. And so, kind of answer this, but why would we want to trim filter the raw sequence data? Unmute your mic, put it in the chat. Oh, I don't know if you want to share that link with everyone. I could do that. So Elba just shared a link about Illumina's pattern flow cell technology. Why would we want to trim or filter our raw sequence data? Cut out any adapter sequences, right? To not confuse mapping, remove adapter sequences. So we won't be removing duplicates in the trimming, but that's something that we could do. You do not need, there are some trimming tools, Susie, where it can remove duplicates that way in the trimming step. Chief, you do not need everything, just what you're interested in. What do you mean by that? You can unmute your mic.
Oh, just that you don't really need all the excess I guess code, the line. You just want either the gene you're interested in or just the part of the gene that actually tells you what you want to know. You don't need the beginning or the end.
Why wouldn't you need the beginning?
Because there's probably a lot of I don't know how to put this, but there's probably a lot of unimportant or I guess useless base pairs in the front that just needs to be trimmed. Like they don't really code for anything.
Why would you think that it wouldn't code for anything in the beginning of the sequences?
I mean, I don't exactly remember the reason why, but when I ran the when I did phylogenetic trees way back in my undergrad, when we aligned the sequences, we trimmed it because we were told that the beginning sequences and the ones at the end, they don't they're not really useful for the analysis. We're just interested in like the middle of the sequences. I don't know if that's anything to do with the poly A or the cap or, but it's something similar to that.
I'm curious, I guess Mike, can you respond to that a little bit since you do
Yeah,
micro?
that's just slightly different, Chief. In that case, we're trying to find the most conserved regions of set genes, and sometimes the beginnings or the ends aren't as highly conserved across the different organisms, and they're not as useful for comparison. So it's slightly different than what we're doing here, but that makes sense for doing phylogenetics like that.
But that's after you build your genomes, right? So after you do your assembly to identify which organisms are there that you might
Well,
not.
what Chief's talking about is, yeah, if we already have genes and we're aligning those genes, then we look at that alignment, and there are going to be parts that have bases or amino acids in every sequence that we have. And there's going to be parts that don't have them from some organisms. And that's sometimes better for the alignment and then the tree building part to remove the parts that have a lot of sparsity and that are missing information from different organisms.
Gotcha. Yeah.
So
So
yeah, that's not working at the read level, yeah.
right. So it's more for like genome assembly and after you kind of have an idea of what the nature of those reads are when you put them together. So in this case, we don't know yet what these sequences are. And so all of the sequence, as far as we're concerned, is going to be good to go, came from a gene that we're interested in and knowing the expression, with the exception of, if we look at the position based on our QC data, so we see here, right, at the position at the end of some of the reads, we see an adapter. And so, but you don't see any of that stuff at the beginning. I know we have a little bit of ends in there, so we might want to get rid of those because we don't know what they are. But all that we would really want to get rid of is anything that we added artificially. So our adapters, right, because that didn't come from true biology. And then there are some tools that will remove duplicates at this step. I wouldn't remove duplicates until after alignment, so you make sure that these sequences are mapping to the same exact region. Then you also have to take into account the UMIs We're not going to get into that in the course. It's actually pretty uncommon in bulk RNA-seq to remove duplicates, believe it or not. And you really should only do it if you have those UMIs, because you could be getting rid of true biological duplicates if not. And so some of the reasons here is, which were pointed out by some of you guys, is to remove low quality reads, right? So we did see that there was a drop off in the quality at the end of the read, and some of them dip below into like poor quality. So we'd want to get rid of those because we're just not confident that that base call is correct. We want to get rid of those adapters that we saw that are at the end of our reads, right? Because again, we added those on artificially. And so we want to get rid of them to increase our mapping efficiency back to our reference genome. Some people trim at the 3' and 5' ends just in general. So some of the reasons for that, as you guys noticed in that end content graph, there was a little blip at the beginning. So some people will trim at that 5' end to account for that. Most of the trimming happens at the 3' end. You could also trim off poly A tails as well if you have poly A enriched data, or if you see a lot of poly A's, you can do that. We don't trim at the 5' end. And then of course, if your reads become too short, right? So if you go ahead and you start trimming your reads, and then there's really poor quality, let's say for example, and you just want to keep on trimming, eventually your read will become really short. And what's wrong with really short reads when we want to map them back to the reference genome? Yeah, and maps to multiple locations. That's right, Ty. So if we have, we trim our reads so much that they become super short, then they're going to map to a ton of locations on the genome, because remember, the genome's highly repetitive, and so then we won't be able to know where it came from anyways. So when we do our trimming and our filtering, after we trim for quality and get rid of the adapters, we do want to assess the size of each of our reads that are left, and if those that are too short, we probably want to get rid of those. And so there are several tools. Are singletons a problem? Would remind me what singletons are, Susie?
They're like reads that only appear once, so they're like entirely unique sequences that could be like some sort of contamination or something.
Oh, that's actually a really good thing. So when we have just one copy of a sequence that we don't have a duplicate of it, then that means it's probably came from a certain transcript. That transcript, to what you might be alluding to though, it might be just lowly expressed. So if we only have one copy, then it could just be a lowly expressed transcript, so we don't see multiple copies of that same sequence. So yeah, that's not a problem in RNA-seq. I'm wondering because anything concerning. Oh yeah, so that's right. A lot of you guys are microbiologists. So Susie mentioned, Mike, that they can be concerning in 16S amplicon sequencing if you just have one copy of a region, and that's because you amplify one very specific region that you're sequencing for 16S. So I don't know, Mike, if you just want to unmute your mic and explain why that's an issue in amplicon sequencing. Probably do a better job than I can.
No, I think you nailed it. I mean, when we're doing amplicon sequencing, that means we're targeting a specific sequence that's highly conserved, a specific fragment of usually the 16S gene. And there's going to be a lot of duplicates because just that's how it works. We're looking at one gene rather than trying to look at everything. And when you have a singleton, something that only shows up once in that case, it's likely to have an error in it because it's very unlikely for an organism to be there with one copy of one sequence and that's not represented by anything else in your data set. So it's just, this is one of those areas where you expect entirely different things based on what you're doing. And another overview statement about FastQC, it's not a problem if any individual module fails. Some of those modules are supposed to fail based on your data. Like you can imagine what we're talking about right now. The sequence duplication levels for amplicon data would be extremely high, and they should be, and that's normal. So FastQC is a general tool, and not anything failing is necessarily a problem. It's always important to look at what your data is and what the module is telling you.
Thanks, Mike. Yeah, does that answer your question, Susie? Great. Good question. Yeah, so it's always important to keep in mind what your original input is that you're looking at when assessing quality, as Mike was mentioning. So now that we know that we want to trim or filter these data, again, to get rid of low quality reads, remove adapters, and then ultimately get rid of anything that becomes too short, there are a variety of different tools for trimming. I should say that trimming and filtering is also referred to oftentimes as pre-processing your data. And some of those tools include cut adapts. Trim Galore is the one that GeneLab uses in our standard pipeline, and Trim Galore actually uses cut adapt under the hood, so it uses the same algorithms as cut adapt. There's also another one called Trim-O-Matic. HT Stream is another one built by Matt Settles at UC Davis, and actually the HT Stream one you can remove duplicates if you want to at this step with that tool. So we use Trim Galore. All of these tools pretty much work pretty similarly, so really you're not going to have too much difference if you like trim with cut adapt versus Trim-O-Matic, so long as you use the same settings for your quality trim and you identify the same adapters. So pretty much you can use any of them. We use Trim Galore because it's actually a wrapper, so it has a bit more functionality. You can actually run fast QCs directly with Trim Galore if you wanted to, and so we use that one. Just like when we were doing our Unix BB tool, yeah, BB Duck is another one, right, Rachel? Yeah, BB Duck and BB tool, those ones are also used quite frequently in metagenomics and Amplicon. Trim Galore, just like all the tools that we used when we did our intro to Unix and also when we did intro to R, the tools come with different options that you can use and you can set. All of these tools that we will have a set of default options. So if you don't know too much about the tool that you're using, of course, I would recommend that you read the documentation for that tool to understand what that tool does and the different options available. But if you're unsure what you should put for a certain value of an option, it's usually good to just start with the default that's provided by the tool. And then once you get to know your data a little more and you get more comfortable with the options, then you can kind of tweak it to fit your needs. So for instance, for Trim Galore, it doesn't, for example, trim at the 5' end by default. And so if you were to take a look at your raw data, your raw fast QC analysis, and if you notice that there's like a real lot of ends at the 5' end, so we had about 1%-ish, which is not bad at all, but if you had like 50-60% of your first couple base pair is ends, then you might want to add an option in Trim Galore to just cut off the first 5 base pairs at the 5' end. So you can kind of play around with this and tailor it to your needs based on what you see. Again, we use the same settings for all of our RNA-seq data. And so I just want to go over a couple of these options that we'll be running. So for Red 33 is going to instruct cut adapt to use the ASCII 33 quality scores. So those are the quality scores provided in your FastQC files. So a while ago, earlier on in sequencing, there was a Phared 66 was used. And so you can also put a Phared 66 in here, again, depending on what the quality scores are in your data. So we know that our data uses ASCII 33, and so we use the Phared 33 option here. That's also the default. We want to tell Trim Galore what adapters we have. And so we have Illumina adapters, and so we use the -- Illumina option, which will define the adapter sequence to be trimmed is the first 13 base pairs of the Illumina universal adapter. This is the sequence. Oops. That's the sequence of the Illumina universal adapter. So Trim Galore algorithm is going to look for that sequence. It'll actually look for any base pair in that sequence and it'll cut it off. So this is going to become important. So anytime so we do a 1 nucleotide match. And so as long as it matches even just 1 nucleotide, then it will cut it off because it'll assume that it's an adapter. We can also indicate what quality we want to use for our cutoff. So those Q scores. And so you want to get rid of those low quality reads at the end. So if it's not defined, the default is to use a 20, which is a 99.9% accuracy. And so we actually use the default. So we get rid of any base pair from the free prime end that's less than a Q20. you want to indicate which length you want to completely remove a read. So again, as you go through and you remove the adapters and you remove base pairs if they're low quality, you can start cutting down your reads so much that it gets really small. And as Ty pointed out, we don't want really small reads because they'll map to multiple locations on the genome. And so we can set and say, hey, if the read gets less than this many base pairs, then we just want to get rid of it. And so the default here is 20 base pairs, and we actually, we use that default. So pretty much as it's going through and doing the quality trimming, if it's left with less than 20 base pairs, then it'll just remove that read from your FASTQ file. And then we also want to let the algorithm know if it's paired or not. So paired end reads, we would have to add this dash dash paired option, and it indicates that read are paired end. And so that tells it that both the forward and the reverse read has to pass the threshold in order to be kept. And so the reason for this is when you use downstream tools, if you tell those tools that there's paired end reads, it's going to look to make sure that you have a forward and reverse read for every sequence that's provided. And so if you happen, and remember I mentioned that the reverse reads tend to be slightly less quality than the forward reads. So you might end up having to throw away more reverse reads than forward reads. But if you throw away a reverse read, you'll have to throw away the corresponding forward read, or else you'll get errors in your other tools, because when you say paired end, it expects it to have a forward and reverse read. Questions about that? Okay. So once we trim our data, we are going to assess the quality again. And so this is the last thing I'm going to go over before we hop into the Jupyter notebooks and actually do this stuff on our data set. And so once we go through and do the trimming with Trim Galore, then we'll have one of the outputs is, of course, a new set of FASTQ files for each sample. That new set of FASTQ files should contain your cleaned up data, your cleaned up reads. And so what you want to do now is assess the quality again of your trim data to make sure that you accounted for some of the stuff that you saw when you did the quality of your raw data. And so this is just looking at the basic statistics. Again, this time it's that same read that we were looking at the raw data QC for, but this time it's after trimming. So after we trimmed with all the settings that we just went over. These are now the statistics. And just for some comparative analysis, I'm going to pull up those same values from the raw data. And I want you guys to take a look at how these values have changed in the trim data and answer some questions. So the first one is how many reads were removed during the pre-processing. So how many reads were removed when we did the trimming? $200,000, around 150K, 150K-ish. Yeah. So the 150K-ish is the right answer, and how we get that is by looking at the total sequences, right? So one of the basic statistics provided by the FastQC algorithm is it tells us how many sequences are there. And so we see that in our raw data, remember we had a sequencing depth or total number of sequence of just over 100 million. But now after we trim, well, we had 100,600,000, and after we trim, our total number of sequence is now 100,450,000. And so to know how many reads were removed after trimming, it would just be the ones that we started with in the raw data minus how many we're left with after we trim. And so it's 146,666 reads that were removed. So yeah, roughly 150,000. What's the sample read length after trimming? Yeah, Susie Mark, that's right. So we see that right here in sequence length. Notice now that after we trim, there's a range. And the range is from 20 to 150. Why does it go from 20 to 150? Why is that the range? You guys unmute your mics, put it in the chat.
Isn't it to cut out the polymerization in the beginning of it?
What do you mean by that?
Because it showed on the graph before that at the beginning there was an uptick on the at the beginning of the read, and
Yes.
I thought I remember you saying that it was because of the polymerization or that there were primers at the beginning of it. So
Yeah.
I was just assuming that they cut that out at the beginning, so it was between 20 and 150 base pairs.
So you can cut that out. We don't actually. So we don't do any 5' trimming. So that comes at the 5' end. So we only trim from the 3' end, but you can set your trimming algorithm to trim at the 5' end because it was like less than 1% again of the reads that were sampled in FastQC that had that N at the beginning. That's not too concerning. So we don't actually trim from the 5' end. So we only trim from the 3' end. And then if you remember, when we sequenced this originally, the total length at the start was 150, right? So the fact that the max range that we're seeing now in the sequence length is 150, it's because that means that there were some reads that didn't have to get trimmed at all, right? So they started at 150, they didn't have to get trimmed because they met the quality cutoff, they didn't have any adapters in it. And so we're going to have some of those reads that remain 150 because the quality is good all the way across and because it didn't have any adapters. Our low end is 20. And so that means that the shortest read that we're going to have in our FASTQ file now is going to be 20. Why does it stop there?
Because that's the shortest it can be without getting sequenced with a bunch of other places.
That's true, but specifically, we set that, right? So in this length option here for trim galore, we tell it how long that we'll say, okay, we can keep it if it's at least this length. And so if you don't say anything, there's a default of 20, and we actually use the default of 20. So we tell it if it's at least 20 base pairs or longer that we could keep it. If it's less than 20, we want to throw it away. We want to get rid of it. And so that's why now the smallest read length that we have is 20 base pairs.
Okay, that makes sense. I just completely went over my head. Thank you.
Yeah, no problem. So that's why we have that specific range. I now want to take a look at what the quality is like now. Yeah, Susie, you chose that. So we want to look at what the quality looks like now after trimming. And so just to compare it with our raw data, so some of the things to notice here, I probably prompt you some questions. So how was the per base sequence quality changed after trimming? What do you guys notice about the quality before and after? Does it improve? What location does it improve on? Yeah, it's better. Specifically, it's better at this 3' end. So again, as I mentioned, we don't actually trim at the 5' end at GeneLab, although of course you can if you so choose. And one of the reasons we don't is just originally in the raw data, right? If you take a look at the position at the 5' end, although we do have a little blip of ends. Overall, the quality at the beginning of the read is pretty good, right? It's in this Q30 or higher, so it's over 99.9% accurate. But what we did see happen is at the end of the read, at the 3' end, we saw some outliers here dip into this poor quality range. And so again, if you remember, we used the default cutoff value for quality. And so the default cutoff value for quality for trim galore is a Q20. And so anything that has a quality score of 20 or better is going to be kept. Anything that has a quality score of less than 20 is going to be chopped away. And so what we see here is now when we take a look at the 3' end of these reads, all of them that are remaining are going to be above that Q20 threshold that we told it to use. So yeah, it improved. So what would happen to the quality if you used a more stringent Phred score cutoff? So if you use something higher than 20, if you use 28, for instance, what would happen to the quality of your trim data? Yeah, it'd go up even more. So instead of having a Q cutoff of 20, if we said we want everything a Q of 28 or higher, then when you go and you assess your quality after trimming, you wouldn't see anything go past a Q of 28. So we say at least a Q score of 20 is going to be plenty good enough to be confident that those bases are accurately called. And so that's why we use the Q20. It's also the default, and that's really good quality. But you can make it more stringent if you so choose, again, by setting those different options in your trimming parameters. So yeah, post-trim video have better quality. Adapters, adapter content before we trim and after we trim. What do you guys notice? Last question before we take our break. What do you guys notice about, yeah, 0 adapters, they've been trimmed out, yes. So this was one of the things we wanted to do, right? We told it, we have Illumina adapters, we want you to get rid of those Illumina adapters. And so our trimming algorithm did successfully remove any adapter sequences. So now with our trim data, we don't see any more adapters there. We see it incredibly good quality. And so the question is, would we now be ready to move on to alignment mapping? So looking at the quality of our trim data now, do you think that we would be good to move on to mapping? Yeah. So now our data is looking real good, and we can move on to that mapping step. Okay, so that would be the next step, but we're going to actually do this stuff. So we have a break right now. I want you guys all to be back here in 20 minutes at 9:50 Pacific time. And when we do, we are going to log on to our SMCE accounts and start processing our data. So go ahead, take a break, and please be back by 9:50.